Okay, let's get started because Lizzie has a huge amount of data to present to you today. So I would like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation. I pay my respect to their elders past and present, and I welcome members of other First Nations communities that might be dialing into this talk today or might be present in the room. So what can I say about Lizzie? Well, I've thought about it, and I think in the end I'll come up with a good analogy. So I've been very lucky to have her, and also I'd like to say up front that if you're impressed with her talk and think that the supervisors have done a decent job, then please think of Gemma, who unfortunately can't be here today, because she certainly was a huge part of this. So I became aware of Lizzie when she was working as an honours student with Claire Scott, who's a very close friend of mine. And I heard her seminar at the end of honours, and I thought, she's really technically accomplished. She's clearly very smart and hardworking. And then a couple of years later, after Lizzie has worked as an RA with Claire, when Claire told me that Lizzie is now ready to take the new step and do a PhD, and said, well, she's thinking about going to the Peter Mac, I thought I'll have to rescue her from the dark. And I was very lucky that Lizzie accepted to join my lab. I'm sure the reason that is because I was really smart and immediately made her have a chat with Gemma, who's her co-supervisor, because Gemma is much nicer than me. Coming to think of it, she's also much smarter than me and also a hell of a lot better looking than me, so I don't know what I'm bringing to this party here. When Lizzie joined the lab, Gemma and I had a lot of ideas about what we wanted to do to find out how P53 really functions and what it does, and we thought we need new resources, new mouse models, and other general resources. And we started with the help from the Magic Lab and Marco and Andrew making these resources. And any PhD student would have taken one of these projects, one of these resources, but whenever we had a new idea and said to Lizzie, oh, we're going to make these mice now, she would say, that sounds really interesting. I think I'll look after that as well. And she did, as you'll find out from her talk. So I think the way I can best describe Lizzie is that she's the nicest present that Claire Scott has ever given me, probably the nicest present that anybody in science has ever given me. So over to you.
Thanks for that kind introduction, Andreas. It's my pleasure to stand here today to talk about the work that I've been doing in my PhD on the tumour suppressor gene P53. So P53 is a transcription factor that directly regulates around 500 target genes. P53 mutations occur in around 50% of cancers, mostly in the DNA binding domain, and up to 90% of cancers have defects in the P53 pathway. People with Li-Fraumeni syndrome are born with one mutated and one unmutated copy of P53 and have an increased risk of developing cancer. It's around 50% incidence by the age of 35. So from all this, you can see that P53 plays a clear role in preventing cancer development. P53 can be activated by several processes that cause stress in a cell. These triggers are things like nutrient deprivation, DNA damage, and oncogene activation. In an experimental setting, we can also use the small molecule called Nutlin-3A to activate P53. Nutlin is an MDM2 inhibitor, and MDM2 is the E3 ubiquitin ligase that is responsible for the turnover of P53 protein, which is usually kept low in an unstressed cell. So when we inhibit MDM2, we get stabilisation and activation of the P53 protein. So when P53 is activated, it can cause a number of different cellular responses. These can be things like changes in metabolism. It can cause a cell to undergo cell cycle arrest or senescence. It can cause a cell to undergo apoptosis, or it can help with DNA repair. And a particular cell will tend to only follow one of these responses. And we know that there are certain genes that are critical for the regulation of these processes. For cell cycle arrest and senescence, this is P21. And for apoptosis, this is Tumor and Noxa. And these are the three cellular responses that I'll be focusing on today. So currently, it is thought that if after P53 is activated, a cell upregulates Tumor, then this will cause a cell to undergo apoptosis. Alternatively, if a cell activates P53 and then upregulates P21, this will cause a cell to undergo arrest. But despite 40 years of research into how P53 functions, we still have a few major questions left to answer. Firstly, what is the relative contribution of these pathways for P53-mediated tumour suppression? Secondly, what is the most effective way to harness P53 activity for anti-cancer therapy in tumours with wild-type P53? And thirdly, how can we treat cancers with mutant P53, as these have a poorer prognosis than wild-type? So today I'll be telling you about our work trying to determine which factors dictate cellular outcome after wild-type P53 activation. And then I'll take you through a series of new models that we have created to help answer these questions I posed before. So we know that after P53 activation, a cell will tend to follow only one P53-mediated outcome. But what dictates this is unclear. So to answer this, we first needed to understand how the cell types responded to P53 activation. So I took 6 cell types from mice, so fibroblasts, bone marrow-derived macrophages, breast organoids, thymocytes, neurospheres, and pre-B cells, and treated them with Nutlin-3A to activate P53, and then asked what cellular outcome was dominant. So most assays only determine 1 or 2 outcomes. So we needed an assay that would be able to simultaneously measure all of these outcomes that we were interested in. So during my PhD, I developed a FACS assay that works on both human and mouse cells to overcome these limitations. And in this assay, to look at cell cycle arrest, we use BRDU incorporation to look at DNA synthesis, and 7AAD to look at DNA content. For senescence, we look at a cell surface marker, UPAR or CD87, which accumulates on senescent cells. And for apoptosis, we use Nexin-5, which binds to phosphatidylserine residues, which are externalized when a cell is undergoing apoptosis. So I used this assay, and I found that fibroblasts, breast organoids, and bone marrow-derived macrophages all undergo cell cycle arrest. So what you can see here are some FACS plots, looking at DNA content on the x-axis and DNA incorporation using BRDU on the y. And in the red box, you can see S-phase cells, which these cells are cycling. So when I treat them with Nutlin-3A, in the wild type only, we can see that there is a dramatic reduction in these S-phase cells, indicating these cells are undergoing cell cycle arrest. And this might be clearer here, and when you look at the orange bars, which indicate the S phase, that this only occurs in the wild type and not in the P53 knockout cells. So we also found that fibroblasts undergo senescence. So here we're looking at a FACS histogram that's looking at UPAR staining. And so you can see that after Nutlin, there is a shift to the right, indicating these cells are becoming more senescent. And again, we could see that this only occurred in the P53 wild type cells and not the P53 knockout cells. So neurospheres, thymocytes, and pre-B cells all underwent apoptosis. And so this is a survival curve over time, just looking at the proportion of alive cells in the culture. And what we can see is that the P53 wild-type cells treated with Nutlin-3A reduced viability by 24 hours, whereas the control cells, the P53 wild-types treated with DMSO, or the P53 knockout cells did not. So now we can assign outcomes to our P53-mediated, we can assign P53-mediated outcomes to our cell types. So for our fibroblasts, bone marrow-derived macrophages, and breast organoids, this is cell cycle arrest. And the MDFs also showed signs of cellular senescence. Thymocytes, neurospheres, and pre-B cells, these all underwent apoptosis. And so as P53 is a transcription factor, the next logical step to us was to perform RNA sequencing at an early time point to see what was, to investigate what was changing and try to determine these factors that dictate these cellular outcomes. So to do this, I took 3 wild type and 3 P53 knockout mice, and I isolated these 6 cell types from each of them. And then for each cell type, I would treat some with DMSO and some with NUTLN3A. And then I extract RNA, and then we sequence with the help of Stephen Wilcox. And the following bioinformatics analysis has been done by Pedro Baldoni and Gordon Smythe. And this is not a small analysis in any way. And the data that I'm just showing you is only the beginning of what we can find from this data set. So I really want to thank both Pedro and Gordon immensely for all their hard work and advice on this project. So firstly, we performed some quality control, just looking at things like P53 levels in the samples. And then for each, then the next thing we wanted to do was for each cell type individually, look at the differentially expressed genes that occurred between the DMSO and the NUTLN3A treated samples in the wild type cells, and then also in the P53 knockout cells. Then we initially had 3 questions we wanted to answer. Firstly, how similar are the differentially expressed genes across all our cell types? Secondly, can we identify our pathways of interest in our cells? And thirdly, which differentially expressed genes in these pathways are direct P53 target genes? So this is an example of one of the cell types, fibroblasts. So here we're looking at plots that show the differentially expressed genes that occurred between the DMSO and NUTLN treated samples. On the left, we have changes that occurred in the wild type cells, and on the right, in the P53 knockout cells. And as you can see here, there was very few change at differentially expressed genes occurring in the P53 knockout cells, indicating that NUTLN3A was quite specific at only activating P53 targets. And you can see that there was many more differentially expressed genes in the wild type cells. And when you look at these changes on a heat map, you can also see that the P53 knockout cells and the DMSO-treated wild-type cells display similar expression patterns, with only the wild-type cells that have been treated with NUTLN3A displaying this P53 activation profile. So our first question was, how similar are the differentially expressed genes across all our cell types? So to do this, we used a Pearson correlation of the Z-scores of the differentially expressed genes in each cell type. In this plot, the darker blue colour means there is greater correlation. And I've colour-coded the cell types based on the cellular outcome for the next few slides. Green for cell cycle arrest and orange for apoptosis. So looking at this plot, we saw that there was a lot of similarity in the changes that occurred in the blood cells that undergo apoptosis. On the other hand, there was also a lot of similarity between the 3 cell types that underwent cell cycle arrest. Interestingly, our neurospheres were most similar to the fibroblasts and the other cell types that underwent arrest, even though that they undergo apoptosis. Because of this cell type might be crucial to understanding which genes are the most important for when we start looking at the individual genes. So next we wanted to see if we could identify outcomes of interest in our cells, so apoptosis, cell cycle arrest, and senescence. To do this, we can look at pairwise correlation plots filtered down to the genes listed in the KEGG pathway for that outcome, and that were also differentially expressed in at least one of our cell types. So here, the red dots on these plots represent senescence pathway. What we can see is that there are strong relationships between the MDFs and the other cell types that are arrested. And in cell types that undergo apoptosis, we saw a lot of downregulated genes. And in cell types with different fates, there was little correlation. Next, we looked at the cell cycle arrest in the same way. So the red dots now are the KEGG cell cycle pathway genes that were also differentially expressed in our cell types. Again, we could see strong relationships between fibroblasts and the other cells that arrested. And again, with the thymocytes, we saw a less good correlation. And in the cell types with different fates, there was even less correlation. And finally, we looked at the apoptosis pathway. Now the red dots are the genes listed in the KEGG apoptosis pathway that were also differentially expressed in one of our cell types. So here we could see a strong relationship between the cell types that undergo apoptosis, a weaker relationship in the cells that that underwent cell cycle arrest, but an even weaker correlation in the cells that had different outcomes. So the take-home message from the last few slides is that we can see a pathway signature in our cells that corresponds to the ultimate outcome of the cells. But this is looking at the pathway as a whole and not just P53 direct effects. So does this mean we are just finding that we can predict the outcome that these cells will ultimately follow, but not the driving factors behind this? So we thought if we narrowed down these lists down to direct P53 target genes, we might be able to answer this. So the first thing we noticed when we started looking at the lists of differentially expressed genes in each cell type was that there was 114 commonly differentially expressed genes across all cell types. This is represented by the heat map seen here. And these genes included cyclin G1 and P21 that are involved in cell cycle arrest, MDM2 and MDM4, which are involved in the negative feedback regulation of P53, and PUMA, NOXA, and BAX, which regulate apoptosis. And this was already exciting to see, because if PUMA and P21 are both increasing, this means they are not the sole reason for a cell following one particular outcome. And interestingly, when we looked at this pairwise Z statistic plots again, but this time looking at the genes listed in the KEGG P53 signaling pathway, now despite ultimate outcome of the cell, we can see a strong correlation in gene expression. But this still just shows us that we have P53 pathway activation. So next we looked at the P53 direct target genes. Now these plots show the red dot. Now in these plots, the red dots are direct P53 target genes. And this list of genes was made a few years ago by Anna Voss from what was based in the literature. So now we can see that the cell types that undergo the same fate show a stronger correlation, and the cell types undergo different fates. So next, we wanted to limit the direct target gene list down to just the genes in our pathways of interest, so cell cycle arrest, senescence, and apoptosis. And this is fairly new analysis, but it seems like this might not tell the whole story, as the apoptosis genes in red increased in all situations, as do the purple cell cycle arrest genes and senescence genes. So our next step was to look at one pathway in detail. And as our lab has a lot of expertise in apoptosis, and this is a P53-mediated outcome we are very interested in, this made a logical place to start. So a cell commits to apoptosis when the pro-apoptotic proteins BAC and BAX are activated. And this is the point of no return. BAC and BAX can be restrained by the pro-survival proteins BCL2, MCL1, and BCLXL. These themselves are inhibited by the BH3-only proteins BIM, PUMA, and NOXA. And so when P53 is activated, it can increase these levels of the BH3-only proteins and start the cell on a path to death. Now, whether or not a cell will die is thought to be due to the levels of these pro-survival proteins and these pro-apoptotic proteins in the pathway. So if the pro-survival proteins are in excess, a cell is likely to survive. And if the pro-apoptotic proteins are in excess, this would push a cell to death. So we went ahead and looked at where all these proteins, all these transcripts now, were in our cells. So here we can see two cell types that underwent apoptosis. And we could see that NOXA, PUMA, BAX, and BIM were all upregulated in both these cell types. But the pro-survivals were not. When we looked at two cell types that underwent cell cycle arrest, we could again see that PUMA, NOXA, BIM, and BAX, the pro-apoptotics, were increased, upregulated. But this time also we could see that BCLX and MCL1, the pro-survivals, were now also increased. And this was pretty exciting because we thought that this would is likely to change the balance in this pathway and potentially be the reason for these cellular outcomes. So we just looked at 4 cell types there, so then we wanted to look at all 6 of our cell types. And so what we saw in all 6 was, again, that the pro-apoptotic genes increased in all of these cell types regardless of outcome. And when we looked at the pro-survival genes, these increased in cell types that underwent cell cycle arrest. However, the neurospheres, like in the other analysis that I've shown, are again an outlier to this trend, so it'll be very interesting to delve further into this cell type and analyse these genes more. So we think this is pretty exciting. And so currently our hypothesis is that if a cell upregulates P53 and then PUMA, NOXA, and BAX, this would lead a cell to undergo apoptosis. But then if you have a cell that upregulates P53, PUMA, NOXA, and BAX, and then MCL1 and BCLX, this would cause our cells to undergo cell cycle arrest. And so we want to follow this up. And so one way to do this would be to take a cell type that undergoes apoptosis usually and see if we can increase the MCL1 and BCLX levels and see if this would cause the cells to undergo cell cycle arrest. Alternatively, we could take cells that usually undergo cell cycle arrest, decrease levels of MCL1 or BCLX and see if this induces cell death. And we could do this either with gene knockouts or using the BH3 mimetics. So the next thing that we wanted to look at was the dynamics of pathways of cells following P53 activation, because we know our RNA sequencing is only performed at one time point. And we know that cellular outcome is a dynamic process. So we needed new tools to look at the timing and extent of upregulation of these genes that determine cell outcome after P53 activation at a single cell level. And then we wanted to know, when do cells commit to their outcome, and do cells in different niches act differently in vivo? So we know what genes are critical for P53-mediated apoptosis and senescence, and this is Puma and P21. So to be able to answer these questions, with the help of the Magic Lab, so Andrew and Marco, we created some P53 response reporter mice. So to be able to follow the cells undergoing apoptosis, we created a Puma TD tomato mouse. And this involved replacing the coding sequence of Puma with the coding sequence for tomato. And as we have essentially removed one allele of Puma by doing this, we always use these mice as heterozygotes, so they will still have one allele of functional Puma. The second strain we created was to study cell cycle arrest and senescence. So here we have left the P21 gene intact, and then afterwards inserted an IRES, so an internal ribosomal entry site, and the GFP coding sequence, so that when P21 is transcribed, GFP is also transcribed. So first we needed to answer two questions about these mice. Firstly, by inserting these fluorescent reporters, have we changed how these cells, proteins function? And secondly, can we detect our reporters? So I took some thymocytes from our Puma tomato mice and tested if they would still undergo apoptosis. And so again, this is just death assays looking at survival over time after different stimuli. And we can see that the Puma tomato thymocytes behave similar to the Puma plus minus thymocytes. I also isolated fibroblasts from these mice and looked to see if they would undergo cell cycle arrest. So here we have some DNA content plots, and overlaid are a DMSO and a Nutland-treated sample. And the first peak indicates the cells that are in G0 or G1. The little dip indicates cells in S phase and are cycling, and the second peak indicates G2M cells. So what you can see in a wild-type mouse is that after Nutland 3A treatment, there is a decrease in the S phase cells. And we could see this in our Puma TD tomato mice, cells from our TD tomato mice. And this is not, and as we are using Nutland as the stimuli, this was not seen in a P53 knockout. So next we wanted to see if we could detect our reporter by fax. And when we looked at untreated thymocytes compared to wild-type thymocytes, we could already see some level of tomato expression in these cells. So next, I treated these thymocytes with Nutland 3A, and we could see an increase in our reporter colour expression. And this didn't occur on a P53 knockout background, indicating that we can at least do this in a P53-dependent manner to activate Puma and the reporter. So I also took some MDFs or fibroblasts from these mice, and again, I could see if I treated with Nutland 3A, we could see an increased expression of our reporter. And this did not happen on a P53 knockout background. So we concluded that our Puma TD tomato mice are faithful reporters of Puma expression. And so next we wanted to look at our P21 iris-GFP mice. So I isolated fibroblasts from these mice again, and I looked to see if these cells would undergo cell cycle arrest. And again, we're looking at DNA content plots by fax, And in the wild type, we can see that these cells undergo cell cycle arrest, as do the cells from the P21 iris GFP mice. And this is not seen again in the P53 knockout. So I also took thymocytes from these mice and looked to see if they would undergo apoptosis similar to wild type thymocytes. And again, we could see this was the case. So next, we wanted to see if we could detect GFP expression in these mice by FACS. So I took fibroblasts from these mice, and when we compared them to fibroblasts derived from a wild type mouse, we could already see levels of GFP, indicating that there's a basal level of P21 being expressed. Once we treated with Nutlin-3A, we could see an increase in our GFP colour, indicating that our reporter was being more highly expressed. However, in our thymocytes from these GFP mice, when we compared them to a wild type mouse, we saw that these did not express GFP. And this was even after stimulation using Nutlin-3A and etoposide. So this was quite surprising to us, because we know from qPCR data and our RNA-seq data that the P21 RNA does increase over time in these cells. So we wondered if this could be due to the cell cycle stage that they were in. So I took quiescent T cells and looked at their GFP levels, and I saw that there was very little GFP expression even after activation of the P53. But as soon as I turned these cells into mitogen-activated proliferating T cells, we could start seeing GFP expression in these cells. And furthermore, once there was a further increase in GFP levels after treating with different agents in these cells. So what does this tell us about the control of P21? So, well, we think that maybe in quiescent cells, upon P53 activation, the P21 reporter and maybe the P21 gene are only transcribed and not translated. But we are not experts in gene regulation, so if anyone has any ideas about what is happening, we'd be very happy to hear from them. So our ultimate plan that we want to do with these mice is we want to use microscopy to visualize the cellular dynamics over time. So we tested our P21 iris GFP cells on the incucyte to see if we could detect GFP expression. And what we could, so we had some wild-type control cells that showed no GFP expression. And then from our P21 iris GFP mice, we had some untreated or treated with DMSO and some Nutlin-treated cells. And over the 72-hour incubation period, we could see that the levels of GFP increased in our cells. And this was done with Annabella and Christy. And so now we think that our P21 iris GFP mice are also faithful reporters of P21 expression. So we do want to be able to look at these cells in vivo over time. So we've been talking to Edwin about doing some intravital imaging. And so what this would be to take our reporter mice and some wild-type mice and use gamma radiation to activate P53 in these mice. And then, with Edwin's help, look in the bone marrow in the skull to be able to visualize these cells and how they are responding to this P53 activation. So we did a pilot experiment with these mice and looked at the GFP levels in the P21 iris GFP mice. So over on the left, you can see a wild-type mouse that has been irradiated, and we can see that there's some autofluorescence in these cells. So when we looked at our P21 iris GFP mice, in the non-irradiated samples, we could already see some bright green dots indicating cells that were expressing P21 and the reporter. However, after gamma irradiation, we could see that there was an increase in the number of these cells, and these small green cells in particular. So we were pretty happy that this reporter could be used for this kind of imaging. So we also looked at this in the TD tomato mice. So again, we have a non-irradiated mouse where we could see some low levels of puma reporter expression being expressed in these cells. But after gamma irradiation, there was a lot more bright red small cells, indicating that these cells were upregulating Tumor and therefore the reporter. So what is next for these mice? Well, we want to be able to measure multiple P53 activated pathways simultaneously. And so we want to know, do both of these reporters turn on in the same cell? So we're crossing these mice together. And we then want to know at what point do these reporters get induced? And we want to monitor fate of cells that have turned on one or several of these reporters. And we also have additional reporter mice looking at P53-mediated DNA repair and metabolism, and this is Annabella Thomas's PhD project. And so we've just started crossing these mice together, but I do have one little bit of exciting data to show you on this. So this is very preliminary, but what we can see here is that from these crossed mice, we have isolated some fibroblasts and either treated them with DMSO or Nutlin for 24 hours. And what we can see by these FACS plots is that almost every cell is upregulating both GFP and TD tomato. And this was just an amazing result for us. So this would mean that regardless of cellular outcome, these key genes for these distinct outcomes are being turned on in the same cells after P53 activation. And so now we're really looking forward to the future possibilities that we can do with these mice. So the next part that I'm going to talk about is these P53 flag mice that we have developed to identify direct transcriptional targets of wild-type P53. So we know this would be very interesting to correlate with our RNA sequencing results that we showed before, because these direct P53 target genes seem to not be acting as we might have thought they were. So our ultimate goal is to perform chip or cut and run using for P53, but currently the antibodies are not particularly good for this. So to help with this, we decided to create an N-terminal P53 triple flag tagged mouse, and this is a mouthful to say, so it's the flag tag, P53 flag mouse. And this was created again by the Magic Lab, so Andrew and Marco. And so what they did was insert a triple flag tag into the endogenous P53 loci. And so any P53 protein from this mouse would also include this flag tag, which we could then ideally use flag antibodies for instead of P53 antibodies. So here is a Western of some MDFs that I generated and then treated to activate P53. And we were looking to see if we could see P53 expression. And what we could see that was in a wild type unstressed state, you usually don't see P53 on a Western blot. But once it's stabilized, after you activate it using Nutlin or irradiation, you usually start to see a pretty strong band. And we could see that in our P53 flag mice, in the homozygotes, this band was shifted up, indicating that we had incorporated the flag. And in the heterozygote, we could start to see two bands. And we could also see that we could use a flag antibody to detect the flag expression in these cells. But we needed to make sure that we hadn't disrupted P53 function by inserting this flag tag. So I generated MDFs from these mice, and I looked to see if they underwent senescence the same as wild-type MDFs. And we did find that this was the case. I also took thymocytes from these P53 flag mice and looked to see if they would undergo apoptosis similar to wild-type cells. And again, these cells did what we expected. We also found that upon P53 activation, cells from these P53 flag mice can induce P53 targets, genes such as P21 and Tumor, normally, which I did through qPCR, but I'm not showing you the data today. But most importantly, we found that the P53 flag mice don't develop cancer. And this is of particular importance because we know that all mice that have complete loss of function of P53 develop cancer before 300 days. So we think these P53 flag mice are fully functional. And so we're getting ready to perform cut and run on cells from these mice to identify the P53 direct transcriptional targets. So the final part of my talk is on the role of mutant P53 in the development and sustained expansion of cancers. So I mentioned at the start of my talk that people born with one mutated copy of P53 have a disease called Li-Fraumeni syndrome and are susceptible to cancer development throughout their lifetime. Here we can see a pedigree of one such family, with all the affected persons developing cancer before the age of 40. We also know that cancers with mutant P53 are often harder to treat, so part of what we want to do is investigate therapeutic options for these cancers. So as we have been talking about so far, wild-type P53 works as a tumour suppressor by activating a number of cellular processes. However, when there is a point mutation in P53, this can have a number of different consequences for a cell. One consequence is that there is complete loss of wild-type function of P53. Another is the mutant P53, and if the wild-type is still present, will bind together, and the mutant can then modulate the wild-type function, causing these dominant negative effects. And the final consequence are gain-of-function effects. And this is where mutant P53 is thought to bind to other proteins, such as other transcription factors, which can then induce expression of genes that are not controlled by wild-type P53 and might regulate different processes, such as genes that could drive metastasis. But these gain-of-function effects are still being debated whether they exist and what role they actually play in. So, depending on whether or not a tumour relies on these proposed gain-of-function effects for survival will ultimately dictate what therapeutic options are. So, if gain-of-function effects exist and are crucial for tumour cells, removing mutant P53 from the cell should work to kill the tumour cell. So, we need to create tumour models in which we can remove mutant P53 and turn them to a P53 knockout state. If these gain-of-function effects are not critical, though, we will need strategies to be able to restore wild-type P53 function. So to test this, we would need tumours in which we can remove mutant P53 and restore the wild-type P53 expression. And another option could be to bypass mutant P53 and activate these downstream tumour suppressive functions. But this would only work if the downstream pathways are still intact. And we could do this by using certain drugs. So to help us answer these questions, we developed two switchable P53 mutant mouse strains, and this again was done by the CRISPR lab, so Andrew and Marco. So I'll take you through how these mice work. So this is the first mouse, and it is a wild-type to mutant to wild-type mouse. So up here we can see the DNA. And what we've done is inserted two different P53 coding sequences into the endogenous allele of P53. The first is just a wild-type coding sequence. So at birth, these mice produce wild-type P53. As this is between fret sites, we can come in with a flip recombinase. And once we do this, this would excise this first insert, and this would lead to expression of our second insert, which is a P53 mutation. This is between locked P sites, so we can come in with a Cre recombinase, and this would remove the second insert and restore endogenous wild-type P53. The second mouse strain is a wild-type to mutant to P53 knockout strain. So this starts the same as the previous one. We've inserted two different coding sequences of P53, but also this third locked P site here. So at birth, these mice express the first cassette and so are expressing wild-type P53. When we come in with a flip recombinase, we can remove this first insert and lead expression to, and have expression of our second insert, which expresses the mutant. This is between LOX P sites, so we can come in with a Cre recombinase, and this time, as we have this third LOX P site, we'll remove the second insert, but also endogenous exons 2 to 6. So this would lead to a non-functional non-functional protein that would probably be degraded by nonsense-mediated decay. And so by using these two mice, we can pull apart the role of these gain-of-function effects of mutant P53. So to validate these mice, I made MDFs from them, and I looked at the P53 levels, expression levels on a Western blot. So as expected, in these unstressed wild cells from birth, which have wild-type P53, in the wild-type to mutant to wild-type strain, these didn't have detectable levels of P53 on our blot. When we came in with the flip recombinase, we could start seeing expression and stabilization of P53. When we introduced a Cre recombinase, we could see that the levels of P53 in these cells decreased, but were not restored back to the wild-type levels. And this could be that we hadn't had complete removal of the P53, or it could be that our P53 is now activated wild-type P53 and stabilized. So I also looked at the levels of P53 in these MDFs from the wild-type to mutant to knockout mice, and again, initially these had non-detectable levels of P53 on a blot, and after flip recombinase we could see stabilization of the P53 protein, indicating that we had induced a mutant. This time, when we came in with the Cre recombinase, we could see complete removal of the P53, indicating that we had achieved a P53 knockout state, and also probably suggesting that on our first blot we had induced complete removal of the P53 as well. So we do plan to use these models in a heterozygous state and introduce the mutant in only a few cells using a viral delivery of a flip recombinase, like how we believe cancers would usually arise in a person. However, to validate this model, that this model didn't induce tumours, we decided to test this in a kind of sledgehammer system. So to do this, we crossed our mouse to a constitutive flippy mouse, so all offspring would express P53 mutant from conception. And then we could cross these together further to get homozygotes. So I've said before, and now this is data showing this in the pink, that all P53 knockout mice succumb to disease before 300 days. And P53 wild-type mice remain tumour-free throughout this period. When we made a survival curve for our P53 homozygote mutants from birth, we could see that this overlaid with our P53 knockouts, and they all succumbed to disease before 300 days. So this was pretty promising. And the P53 mutant heterozygous mice were also falling sick with tumours before the wild types. Now, I saw that tumours arising in these mouse models expressed high levels of mutant P53. And this is just P53 IHC showing this. So we know that P53 mutations are prevalent in lymphoma, and it's even more so in this relapse setting. So we went about creating a lymphoma model. To do this, we crossed our mouse, our switchable P53 mutant mice, to EU-MYC mice. And these spontaneously develop lymphomas that are accelerated by P53 mutations. And so when we did this, we saw that our switchable P53 mutant also accelerated lymphoma development in these mice to a similar extent seen by EU-MYC P53 plus minus mice. So we wanted to ask, do these lymphomas driven by this mutant P53 rely on the sustained expression of this mutant protein for continued expansion? And we also wanted to ask, would removal of mutant P53 and restoration of wild-type P53 be a therapeutic strategy? And this is the question that I have a bit of data for today. So the way that we wanted to test this was that I generated some lymphoma cell lines from this EU-MYC switchable P53 mouse model. And so I took some lymphoma cells, and then we could either leave some with the Cre unactivated or the Cre activated. And then we could ask the question, do these tumour cells die? And so I would assess the tumour cells, if the tumour cells are dying, by FACS using an XM5 and PI. And the alive cells sit in the lower left quadrant here. And then as they undergo apoptosis, they move across to the dying quadrant and then up to the dead quadrant. So I did this in my cells, and it was really exciting to see that we had a dramatic reduction in viability between our control cells and our reactivated cells in the switchable P53 mutant line within 24 hours of activating the CRE. And so this shows that removal of mutant P53 and restoration of wild-type P53 function induces rapid apoptosis in lymphoma cells. And this wasn't a CRE-mediated toxicity because we had wild-type P53 cells with a CRE and non-switchable mutant P53 cells with a CRE that showed no drop in viability in this. So this was an extremely exciting result for us because this would mean that restoring wild-type P53 function kills lymphoma cells driven by mutant P53, and this could then be used as a therapeutic option. And we've also shown by this that the downstream signaling elements, at least for apoptosis in these cells, are still intact. So the third option, therapeutic option, of coming in with downstream drugs would also potentially work. So we do want to ask, do lymphomas driven by mutant P53 rely on the gain of function effects? And so to test this, I need to perform these studies in lymphomas from the wild type to mutant P53 to knockout mice, which I'm generating currently. And of course, we want to do some in vivo analysis of lymphoma growth after restoration of wild-type P53 function or removal of mutant P53 function and generation of P53 null state to check that my in vitro results correlate to in vivo. And we are expanding these models to look across multiple cancer types. So Tianwei's PhD project involves using these mice in lung cancer with Kate Sutherland. And we also have a number of other cancer models that we are interested in looking into. And finally, these switchable mutant mouse models, as well as the P53 response reporter mice and the P53 flag mice, along with the RNA sequence stacking dataset, are all extremely versatile and valuable tools that will aid in the future of P53 research. So that brings me to the end of my talk. So now I'd just like to thank the many people who have helped me throughout my PhD. So firstly to Andreas and Gemma, thank you for believing in me and taking me on as a student. Thank you for all the support you've given me throughout the last 4 years. And as I can't sum up how amazing you guys are as supervisors in one sentence, I don't think I'm even going to try. You have just built an incredible lab environment with so many wonderful people. So thank you to everyone, past and present, in the Kelly and Strasser labs. Thank you to my PhD committee, Warren, Philippe and Jane, for all your helpful advice on the projects and looking out for me. Also to the BC division, and Herald Lab and the DivCos. So for these projects, I again need to thank Gordon and Pedro for all the bioinformatics and Stephen for the sequencing. For help growing and advice growing the primary murine cell types, I need to thank Anna, Maria, Luke and Marcel for imaging Christy, Edwin and Niall. So a huge thank you to the Magic Lab for generating all these mice. So Marco and Andrew, thank you very much. A big thank you to the bioservices team in room 16 for looking after all my mice. And thank you to all the mice themselves. Bruno, Rainbow and Natalie, thank you for genotyping. So it frees up a lot of my time. And thanks to all the core services and facilities at WeHi, because you're amazing. So finally, thank you to all my family and friends who have supported me throughout this time. Could not have done this without you. And thanks all so much for listening, and I'm happy to take questions if Andreas can figure out how to use Slido.
Thank you very much, Lizzie. We might as well start with a question from inside the room. Ladies first.
So that was really interesting, the analysis of the BCL2 family genes upregulated in the senescence state, including the pro-survival genes, and not in the apoptosis state where you only have the pro-death. So BCLX and MCL1 were up, where you have in the senescent state, in the MEFs. But they are obviously not direct P53 targets. So have you given any thought to how to get at what drives their upregulation in that cell type versus not in the other cell type?
Well, so yeah, we think P53 might have an effect on something that could regulate the levels of BCLX and MCL1. So maybe, although that's indirect, there might be another factor that is direct. So we hope that our P50, our flag cut and run could give us a clue of more of these direct targets. I hope that is answering.
Could you do a CRISPR screen? Could you do a CRISPR screen to turn MEFs into apoptosing cells, not senescent cells?
That would be an excellent idea. Yeah. Thank you.
Actually, highly relevant to Anna's comment and question. A question from Mani, who asked it on Slido rather than in person, and that is, would the explanation why MCL1 and BCLX are upregulated transcriptionally only in the cells that don't die, but not in the ones that die, be due to differences in epigenetic state? So.
Yeah, look, that is a possibility. Very happy to take your advice on that and chat about it at some point, because, yeah, it could be. There's so many possibilities for this.
I recall there was a bunch of papers a while back suggesting that there was a switch between P53 switch between senescence and apoptosis as a result of things happening to the P53 protein itself, like being acetylated. I'm guessing you would have a degree of skepticism regarding those papers.
I don't think we can totally ignore it as a fact. Like, we know P53 can be acetylated, and there definitely are papers that show or suggest that P53 acetylation or stuff will cause a cell to head more down the apoptosis route. But my feeling would be that as it is a transcription factor, we should be able to be looking at this transcription that it does induce and, see.
So and also in your model cells, I'm not really familiar with how all of them behave, but are there some cell types there that that also have a mixture of senescence or cell cycle arrest and apoptosis normally?
So we did find that the bone marrow-derived macrophages showed a significant amount of death. It was like 15-20%, but it was very consistent. So yes, some of the cell types do seem to have a few difference, and it could be that the cell types that die first go into cell cycle arrest before heading on to dying. So yeah, that is totally a possibility.
I'll go to Slido. So one of your PhD committee members, Jane, says, amazing results, Lizzie. Do you have any clues as to why the neurospheres behave as outliers in your large RNA-seq data analysis?
So this is still fairly new analysis, so we haven't looked a whole lot into them. But we were really surprised first at their outcome, that they did actually die. When we first did the assays, we thought they would be a cell type that senesced. So that was the first shock, I guess, but very exciting to see. So we have thoughts that it could be how they grow, or just different levels of these proteins in them. Maybe they just have naturally high MCL1, so we're not seeing a big shift up, low levels. Maybe they just have different levels that are changing this, and so it could be a total amount in the cell that is affecting these cells. But we're very interested to look further into like exactly what differentially expressed genes there are, and learn more about the cell type in general.
Very nice talk. So I'm very interested in your mouse model that you switch the mutant to the wild type. So do you find that after you switch the wild type, P53 is highly stable in the mutant cell, or there's any dysfunction in the DM2 pathway. That's why you can get enough wild type PP53 to induce cell death.
So because these cells switch, so in the lymphomas, because these cells underwent apoptosis so fast, I haven't really looked at the levels of wild-type P53 that we have in these cells. From the Westerns that I did on the fibroblasts, and these cell types would tend to senesce with wild-type P53 instead of undergo apoptosis. I think we were still seeing, I believe that band on the Western is that it's being activated still, so going to cause senescence in these cells. But this is not something that I've looked particularly into yet, and so I can, and that will be interesting too.
So I'll take another question from Slido. It's from Grant, and says P53 protein interactions with other proteins have been proposed almost exclusively based on overexpression studies. Are you going to plan to use your flag P53 mouse model to actually look at that more succinctly and more elaborately?
Yeah, we have many plans for our P53 flag mouse model, and because it's endogenous levels, we do plan. Yeah, we will. Great.
Great talk, Lizzie. I'm interested why you have to have different cell fates. Why, upon activation of P53, do different cell types have these different fates?
That's a good question. I put it down to that to be such a powerful tumour suppressor, it has to have a lot of options to go to do all of its functions. So if it has its hand in all these little pots, it can potentially switch if something goes wrong in the apoptosis pathway, and its cell would usually, maybe it could then freak out and be like, can we go down the senescence pathway today? So I don't know, but it is a very interesting question.
Just to follow up also, your flag P53 mice, it's really surprising to me that no one has done any ChIP-seq analysis on P53 before. Can you comment on that?
So there has been P53 chip done. So I tried to repeat it in our hands, and the advice we got was that there was only one lot of one antibody that was going to work. And even though I tried with that lot, it wasn't looking great. So we thought this would just be an easier way to solve this problem for everyone in the future.
Well, we're right on 2 o'clock, so it remains to thank Lizzie for a fantastic talk, and I'd like to reiterate that really a lot of the credit of the work today you should give to Gemma as well, and of course the Magic Lab and Gordon and Pedro have done an amazing job. It's a great place to work here at WeHi. So thank you very much for attending, and thanks a lot to Lizzie.